We're back and ready for part two of chapter 24 in our microbiology text by Tortora. We'll be talking about microbial diseases of the respiratory system. Um, Typical pneumonias. Bacterial pneumonias is our first section of the chapter. The most common cause, which is therefore called typical pneumonia, it's not just a usage, it's actually a term, typical pneumonia, medical term, is caused by strep pneumoniae. Any other type of pneumonia, therefore, is called atypical, and it could be any other microorganism. We'll talk about a few of them. Excuse me. Lobar pneumonia is referring to when an entire lobe of the lung becomes pretty much taken over by disease, by inflammation and pneumonia. Bronchopneumonia infects the alveoli when it gets always down, all the way down to the microscopic air sacs. That's a different kind of an animal, and really going to cause trouble as fluid accumulates in the air sacs. Pleurisy is when inflammation reaches the linings, the membranes on the surface of the lungs and the surface of the pleural cavities. Those serous membranes become inflamed, and that's called pleurisy. So, Streptococcus pneumoniae, gram-positive, encapsulated diplococci, as you know. There's a bunch of different serotypes of strep pneumonia that cause disease. They infect the alveoli of the lungs, which then begin to fill with fluid and red blood cells, and then the surface of the alveoli is the respiratory membrane. It's lined with blood capillaries, and you need all of that surface area to exchange gases with the blood, to get oxygen into the blood in the short time that's available with each breath. And when you start filling up the alveoli with water, you reduce the surface area, and you're going to now have a difficulty exchanging gases of threatening hypoxia situation in the bloodstream. Diagnosis, there's a chemical called aptokin, which kills strep pneumonia bacteria, and it's not used as a treatment for it, but it's used as a way of testing. Other strep species are not affected by aptokin. Bile solubility test, bile salts kill and dissolve the lysis Streptococcus pneumoniae, but not other strep species. And the presence of capsular antigen in urine can be used to identify strep pneumoniae. Treated with macrolide antibiotics. We don't have as much strep pneumonia in this society because of our use of the conjugated pneumococcal vaccine, the strep polysaccharide capsule conjugated to the diphtheroid toxin. Diphtheria toxoid. Said that funny, didn't I? Got it. So there's some encapsulated streptococci. Haemophilus influenza is another fairly common cause of pneumonia. Gram-negative coccobacillus. Similar symptoms, except that strep pneumoniae produces a rust-colored sputum, whereas Haemophilus influenza has a regular whitish green colored sputum. Risk group, yipes, for this disease, children older and young people. Very often the case with many diseases, very old and very young that are susceptible. But we have a Hib vaccine, Haemophilus influenza type B vaccine, that has reduced the incidence tremendously. It's conjugated in the same way as the pneumococcal vaccine. Diagnosis, one way to diagnose it is culture it, and it depends on X and V factors for growth in the culture medium. So it needs hemoglobin and something else, NADH. It needs to have in the medium in order for it to be able to grow. Treated with cephalosporins. Mycoplasmal pneumonia. Mycoplasma are the tiniest known living organism. They're tiny bacteria with no cell wall. As you know, Mycoplasma pneumoniae causes a relatively mild pneumonia. It's often called walking pneumonia because it's people tend to continue on with their life as if they have just a little bit of a rough cold, and they keep going. Common in children and young adults. Um, don't want to slow down, just want to keep on going. One characteristic, if you grow it in culture, you need to grow it on a special media, but it forms a characteristic fried egg culture. It's funny little, looks like little yolks in fried egg or something as it grows on the culture medium. Diagnosis is by PCR and treated with tetracyclines. Legionellosis. Legionella pneumophila, sorry, let's say that better. Legionella pneumophila is a bacterium that causes Legionnaire's disease, and that was first discovered, I think, and it was in the late 1970s or early 1980s. There was a convention of war veterans from the American Legion in Philadelphia, and all of a sudden, several of the Legionnaires just got sick and died within a short period of time right during the convention. And that caused a complete panic. And so then the sleuths were set about trying to figure out what happened, and they discovered this bacteria growing in the water in the air conditioning system of a hotel. Now we know quite a bit about Legionella. At that time, it was completely unknown, and so it can form biofilms as well. Um, Anyway, so this is a disease that can cause pneumonia, especially in elderly and immune-compromised people. Um, Causes a fever, high fever, and cough. There's another bacterial disease called Pontiac fever that you can look up. We're not going to go into that one. Um, Transmitted person to person by aerosols. Sorry, not transmitted person to person, but by aerosols from again air conditioning and air handling systems from commercial buildings. Is typically the method. Um, Treated with azithromycin and macrolides. Psittacosis. Chlamydophila psittaci is a bacteria, an obligate intracellular parasite bacteria that is associated with bird droppings. So in the city where there's a lot of pigeons and so forth, a lot of feces from birds, guano accumulating, that's when the elementary bodies, the infectious form, are going to be prevalent, and they're going to get into the air and cause inspiration into the respiratory system, hence our topic of today, and cause fever, headache, chills, and disorientation. And in immunocompromised people can be lethal. Um, Diagnosis is with growth of bacteria in eggs and or cell culture and PCR. Treated with tetracyclines to get into those cells. Remember, that's one of the ones that could penetrate into cells and get those intracellular parasites. Um, Again, wait. Okay, changing gears. That just took me by surprise. Chlamydophila psittaci is quite different from Chlamydophila pneumoniae. Chlamydophilium pneumoniae is transmitted person to person. It gave me a little jump there. And anyway, it causes mild respiratory illness, common in young people. Resembles Mycoplasma pneumonia in that it's something that is a little more tolerable than more severe cases caused by strep pneumoniae. Um, Possible association with atherosclerosis. Um, I think that's supposed to be atherosclerosis. Diagnosis with PCR and treating with azithromycin. I'm going to have to look that up. Arthro means of joints. I wonder if there's a relationship there. You can also look that up too. Q fever is caused by Coxiella burnetii. It's one of those weaponizable or considered to be a possible biowarfare agent bacteria. Obligate intracellular parasites, and causes a high fever, muscle aches, headache, and coughing. Um, it can turn into, most of the time it doesn't, but it can turn into chronic disease, and it can ultimately cause endocarditis and heart valve damage years after the initial infection. So that's a problem. That's why Coxiella is considered a big problem, because it has that extra bad tail end to it. And so Transmitted from farm animals, uh, from tick bites, transmitted to humans in the inhalation. So farm animals acquire it by tick bites, and then it goes into the air, gets it gets aerosolized and floating in the air, and you can contract it that way from farm animals or from unpasteurized milk. Drinking unpasteurized milk, get Coxiella if there's infected animals. Treated with doxycycline or chloroquine for chronic infections. Q fever. There's some Coxiella burnettii, and there you can see the endospores forming from these guys. Endospore-like body, I should say. They're not true endospores. They would have to be in the genus Clostridium or Bacillus. The reason this is brought up, it's associated with the afterbirth. The bacteria is prevalent in the placentas of farm animals when they deliver their young, and so you have all these placentas, say on a dairy farm when there's calving, a lot of calving going on. Then you have a lot of the bacteria in the environment, and they form these endospore-like structures, and then that's the way they can get picked up easily by people kicking around the farmyard, and that just shows a picture of the cells growing in the placental cell. Melioidosis, Burkholderia pseudomallii, produces a disease that's endemic in Southeast Asia, northern Australia. It's in moist soils. Gram-negative bacillus commonly affects people with immunocompromise, causes pneumonia or tissue abscesses, can cause necrotizing fasciitis, flesh-eating disease, and severe sepsis. So this can be a very, very serious disease indeed. Transmitted by inhalation, puncture wounds, and ingestion, treated with ceftazidime. So melioidosis. Here's a 27-year-old man with a history of asthma, hospitalized with a 4-day history of progressive cough. After 2 days, spiking fever, gram-positive cocci in pairs, cultured from a blood sample. What could it be? And so we have gram-positive cocci, limits us a great deal. And from what we've been talking about in this chapter, we're looking at Streptococcus pneumoniae. Pneumococcal pneumonia is another name for it because of the characteristic morphology of the cells. And here's a picture of an optoken disc on a plate that's been inoculated to produce a lawn of bacteria, and these strep pneumoniae bacteria are inhibited by the optoken in that disc, and just verifying the presence of that species. There's a little summary table. Once again, these summary tables are great for just zipping through and reminding yourself of all the details about each one of these organisms from our chapter. You can almost take them out and put them into a little folder for each chapter and have them right at hand to study the details. Viral pneumonia can occur as a complication of influenza, measles, and chickenpox. So inflammation just gets into the lungs when these bacteria or these viruses are growing on the epithelia. Few labs are equipped to test, so it's something that kind of goes, people just deal with it. It's not usually diagnosed. SARS-associated coronavirus. There was a coronavirus outbreak, a small coronavirus outbreak in 2003. I believe that was the time that it caused a great stir because people were quite concerned about it. And a person traveled to Toronto, Canada, and with it, and went into the hospital and caused infection of several people before they realized how communicative it was, and they got it under control. And we thought, wow, that's the last we'll hear of SARS because we're going to be real careful about that. Well, we all know what happened in 2020 with SARS-CoV-2, another SARS virus that causes the disease COVID-19. and caused quite a epidemic, plus a lot of social polarization and political polarization around what to do about it. Um, Middle East respiratory syndrome coronavirus, another strain of coronavirus that caused disease in 2012 in the Middle East. So, respiratory syncytial virus, um, it's the most common viral respiratory disease in infants, and is life-threatening in older people. So almost all children get infected by age 2 because it's so common. So causes cell fusion. A syncytium is a cell that has multiple nuclei because it's been formed by the conflation of multiple cells. Um, Coughing and wheezing for more than a week, characteristic of RSV. You probably heard about RSV. It was just this winter. There was a relatively large RSV outbreak in this country that was causing some concern because we still had COVID going around and we had RSV starting to crop up and people were getting nervous about that. But luckily, we've passed through that phase of the season. Um, Palivizumab treatment, interesting. Monoclonal antibody, way to treat this guy. All right, influenza virus. Um, Influenza is a virus that contains 8 RNA segments in a capsid with an envelope. It's an enveloped virus, and it's interesting that it has all those different pieces of RNA that are parts of its genome, because if a host gets infected with multiple influenza viruses or related avian viruses, they can basically shuffle those different RNA segments uh, and cause brand new um, strains of virus to be produced. I forgot the term now; it's escaping me for that shuffling process. It's not the right term, but anyway, they can swap out and create brand new um, 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 combinations of genome segments from different viruses that changes the host range, changes the virulence, and that, along with mutations, can cause a whole bunch of changes in influenza year after year. You never know what's coming down the pike when it comes to influenza in the season. Chills, fever, headache, and muscle aches, and we're talking a pretty debilitating disease. Again, I think a lot of times when people say, "Oh, I had the flu," they really mean they had a cold. Having the flu is a kind of a memorable thing. It's a pretty strong disease. Um, it's a threat to survival, even to very young and very old. Um, there's a little picture of our enveloped virus with all of its pieces of RNA as the genome in there, and the capsid. Um, the couple of the famous elements or components of influenza virus, hemagglutinin, and the so-called hemagglutinin spikes are, by the way, it attaches to its host cells, epithelial cells in the respiratory tract. And also another gene product of influenza is neuraminidase. That's a polysaccharide cleaving enzyme that helps the virus escape from the surface of the cell. Just being budded off. Excuse me. Antigenic drift and antigenic shifts. These things can change relatively rapidly. Um, Minor antigenic changes in the hemagglutinin and neuraminidase form different versions of these viruses, and they're often named that way. H1N1, for example, would refer to a particular specific hemagglutinin and neuraminidase protein. Reassortment is what it's called when they swap out the parts of the genome there. There we go. Um, it allows the virus to escape from the immune system to some degree. As soon as the adaptive immune response gets going, then we have maybe some changes in the coat proteins and the spike proteins. Antigenic shifts is when they reassort and really have big changes genetically in the organism, and that can lead to pandemics, brand new organisms that have not ever been seen by the by the people and animals of the world. Every year, governments of the Western countries especially produce influenza vaccine by scanning. They do surveys to find out what are the most common strains of influenza that are emerging as the most likely ones to infect people in the coming winter. They do this in the fall, and then they produce a vaccine, probably in the summer, and then in the fall, produce a vaccine that can be dispensed throughout the world to try to keep influenza from getting too much mortality in the population. And there's a couple of antivirals that are actually effective with influenza. Relenza and Tamiflu brand names of antivirals. They inhibit neuraminidase so that the viruses can't bud off from the cells and then go on to their host cells go on to infect other cells. Histoplasmosis. We come now to fungal diseases of the respiratory system. So we've done bacteria and viruses. Now we come to fungal diseases. Histoplasma capsulatum is a dimorphic fungus. The yeast form can grow in macrophages and forms lung lesions. Most of the time people don't get particularly sick by it. 0.1% of cases go on to cause systemic disease in normal healthy people. It's acquired from airborne conidia in areas with bird and bat droppings, and it's limited to a certain region of the United States. I'll show you a map in a second, treated with itraconazole. Itraconazole is an important drug that's been relatively recently developed. It's one very strong antimycotic that can cross the blood-brain barrier. People with immune compromise have a tremendous problem with fungi because they're so prevalent, and normal healthy people, healthy immune systems don't get disease from them. But once you have immunocompromised, now you have this massive amount of material in the environment that can make you sick. And here's a little range map showing you where histoplasmosis is likely to be experienced. A second one that can cause systemic disease occasionally in healthy people, coccidioidomycosis, caused by Coccidioides immitis. That's another dimorphic fungus. It's known as valley fever or San Joaquin fever because it originates in the San Joaquin Valley in California, where a tremendous amount of produce is produced for the country. Huge produce farms there in that rich valley among the mountains. Arthroconidia, found in alkaline desert soils of the American Southwest. It forms a sphereole with endospores that form in tissues. Most infections are unapparent, don't cause any noticeable disease, but it can cause fever, coughing, and weight loss. Less than 1% of cases can cause that kind of disease that resembles tuberculosis, treated with fluconazole and itraconazole, again, those modern antimycotics. So there's a little picture of an arthroconidium getting into someone's body and germinating, and then it grows into the sphereole and produces endospores, which then are released from the sphereole and can form cysts in tissues. Here's a little range map for this one for Coccidioides immitis. San Joaquin Valley is here. That's probably where it was first known about prevalence in that area or originating in the area. And then here's the now known region where you'll see a fairly high incidence of that disease. Pneumocystis pneumonia, this is almost like a little insertion here in our chapter because pneumocystis doesn't cause disease in people with a healthy immune system. It's an opportunistic pathogen of people with immunocompromise. It's a primary indicator that someone who has progressed from HIV infection to AIDS, to the acquired immunodeficiency syndrome that results from HIV infection after years of time. Unknown reservoir, possibly humans or soil. It forms a cyst in the lining of the alveoli. The cyst rupture, releasing trophozoites, the eating, growing form of the organism. treated with trimethoprim and sulfamethoxazole. Pneumocystis eurovetzi. There's a little diagram showing the life cycle of this one. Some cysts rupturing out of the spherules forming trophozoites that then divide and go on to form a cyst in tissues, and then more intracystic bodies that will eventually release the mature cysts or release the cystic bodies to go on and form trophozoites. Cyclical effect, and it causes a pneumonia, a life-threatening pneumonia in people with AIDS. Okay, and the third microorganism that can cause fungus that can cause systemic disease in normal, healthy people occasionally is Blastomyces dermatitidis. Causes blastomycosis, North American blastomycosis. So symptoms resemble a bacterial pneumonia, but it also causes cutaneous abscesses and extensive tissue damage in the body. About 100 cases per year, so not a very common disease at all. Treated with itraconazole and amphotericin B, and here you can see the range of where blastomycosis may be acquired. Predisposing factors, immunocompromised state for these fungal diseases. These are opportunistic pathogens. Aspergillus, we said, is everywhere. It's one of the most common fungus in the dirt everywhere you go. Rhizopus, very, very common. Mold and mucor, those are very, very common molds that produce spores. And in people with immunocompromise, those bugs can then get into your body, or spores can get into your body, germinate and cause it. And they can just begin to grow in your body unimpeded because there's no adaptive immune response. And you can actually have aspergillus growing in your brain, mold forming in your brain. Isn't that awful? If you have immunocompromise. All right, 3 weeks after working on the demolition of an abandoned building in Kentucky, a worker is hospitalized for acute respiratory illness. At the time of the demolition, a colony of bats inhabited the building. An X-ray examination revealed a lung mass. A purified protein derivative is tested as negative. A cytological examination of the mass reveals ovoid yeast cells. So what could it be? And you may remember that the first of our 3 fungal pathogens that can cause systemic disease, histoplasmosis, is associated with guano from birds and bats. So that's this story right there. Whoop, I think I skipped something. No? Nope, that's just another summary table there of what we've just been talking about, some of the things we've been talking about in this chapter. So again, those summary tables are really wonderful. You might want to pick all those out and put them together from each chapter and make like a little study guide for yourself, and maybe add in a few things in your notes about it. And that can become a great study tool to make you more efficient getting ready for tests on these subjects. So meet me back here next time for chapter 25, digest.